I'm Jess Gutknecht. I got here probably one of the more recent additions to the department out of the NIC. I arrived in January 2014. I'm 50% teaching and 50% research. The classes I teach are environmental soil biology. I also teach the ESPM transfer orientation class. And last spring I just started teaching one of the Global Challenges courses, the Global Climate Challenge, Creating an Empowerment for Change. And I taught this with a professor from nursing and an adjunct professor who's also a community activist, and it was a blast, and I can't wait to teach it again this spring. Who's in my lab? I have one master's student, Cameron Blake, who will be finishing this fall on the spruce project that I'll talk about more in a minute. I also supervise a postdoc with Peter Kennedy, a lab manager, Gigi, and as others have said, I have several undergraduates either as UROPs or in other capacities in the lab, and they've been really awesome to work with. So what drives me? This is a lot in one slide, and maybe I'm regretting sticking to the template, but that's okay. Everything I do can be summed up in two of my favorite stickers. I love soil and I love microbes. That's about all I need to say. I love thinking about the fabric of life in the soil and how these soil ecological interactions really give rise to a lot of important ecosystem functions that we think about on a community or collaborative or societal level. And another big question I'm interested in is how do soils act for mitigation and adaptation of climate change? And so really, how does this fabric of life relate to these bigger questions that we have interest in? My passion, I love soil and I love microbes, and I'm completely fascinated by how both of them function and form, and I just love studying them. I'm also passionate about the interacting with people. I love interacting with my students. I've started some great collaborations. I'm not going to name the names, but I also enjoy working with different cooperation partners. You know, how do I describe how cool microbes are to someone at NRCS or to a policymaker? Like, that's a wonderful challenge. My goal is, I'm still in the beginning, so to continue building a successful research program where I'm really living these passions every day and really contributing great science to some of our societal issues, be they in human interaction or science communication or whatever. And so that is what drives me. And what do I actually do? These are play out in different types of research projects. So these are three examples. One is looking at microbial carbon. I forgot this one for the microbial carbon use in peatlands under warming and elevated CO2. Microbial growth and functioning under forest biodiversity loss, and climate resilience agricultural management choices. So I'm going to talk just a little bit about my work at Spruce as one example of the types of work that I do. I must be speaking really quickly because so microbial carbon and sulfur use in peatlands. This is the Spruce experiment. You can see the little tiny car for reference of how huge these chambers are. This is in Marcellus terminal forest. And there is warming from 0 to 9 degrees Celsius. So there's a range of temperature treatments, and then this summer they turned on elevated CO2. And what's interesting about the elevated CO2 is that the manufacturer was chosen carefully to produce a strong isotopic signal into the peat and into the system. And then we're analyzing microbial communities with an isotopic tracer approach, both for the carbon use and also for sulfur cycling. And so this is the project and the type of data that we're looking at. This is an example of our C13 data, where less negative numbers mean that it's older carbon, and so you can see we have an older and older signal as we go down in the peat, as we would expect. This peat is up to 1,000 or a couple thousand years old. And so this is our baseline before treatment started, and then we're going to ask, what does this look like in the end? Are we going to see a shift in this with older carbon being decomposed? And equally, we're just starting to get online sulfur isotopes with Ed and Randy of the sulfur isotope like shift also that you're making changes for or making changes. That is all.